All right, so we have a number of folks already, and I'm sure we'll get more in the next few minutes, but we'd like to welcome you all to this IAS-USA webinar. Today's Wednesday, December 14th, 2022. My name is Jose Francisco, and I'm a project manager at IAS-USA. We are excited to cover today's highly anticipated presentation regarding the basics of HIV drug resistance mutations. This webinar will also highlight the recent IAS-USA 2022 update to the drug resistance mutations in HIV-1 released in September 2022 this year. We are delighted to welcome our first presenter, Dr. Douglas Richman, who a distinguished professor of pathology and medicine at the University of California, San Diego, who will go over our introduction slides. Welcome, Dr. Richman.
Thank you. Well, welcome everybody to this webinar. I am really delighted to be sharing this presentation with Bob Schaefer from Stanford University and Francesca Ceccarini Silverstein, who is associate professor at the University of Rome Tor Vergata. We are all three of us members of the mutations working group of the IAS-USA, which prepares the drug resistance guidelines every couple of years or so when new data are merit an update. Whoops. And I'm trying to go forward here. Let's see. Let's do it this way. So the webinar board that organizes these webinars consists of a number of talented members whose financial relationships are designated here. Each of the three of us serves on advisory boards or consults with various companies that are indicated here. So regarding CME, the IAS-USA is accredited to provide CME, and there's 1.5 Category 1 credits of CME available for physicians. And importantly, we welcome other healthcare providers, nurses, pharmacists, and so on. And each of these groups can access the CME for participation in this program. The instructions for claiming credit are on the IASUSA.org website. The educational activities of the IAS-USA require financial support, and we specify that we only accept, we require support from multiple different sources so that we are not indebted to any single funder, and we appreciate the support from the funders that we do have. This, because of the ACCME guidelines, funders may not participate in discussions or ask questions during the webinar. So the on-demand recording and slides for this webinar will be available within 24 hours after the live broadcast on the IAS-USA website. So poll question number one, to make sure that we all know how the process works. Please answer the question. Have you accessed the IAS-USA 2022 update of drug resistance mutations in HIV? And you can have three alternative answers. Yes, no, or you didn't know there was an update. And the answers are evenly split. So it looks like we'll be sharing information with a large number of you. We'll close this and move on. There we go. So you've seen how the poll questions work. To submit questions, please use the Q&A button on the Zoom at the bottom. We hope to be able to address everybody's questions, but if it's a really enthusiastic audience, we may not be able to get to all, and we apologize for that. And one important point is that the chat is available for discussion with other attendees, but if you submit your questions in chat, we will not get to them because we can't handle multiple sources. So please put your questions in the Q&A so we can address them there. So we have 3 pre-test questions, and we will be addressing the information for them during the talk. The first one is, after failing a regimen of tenofovir, TDF, lamivudine, and efavirenz that resulted in resistance mutations in the nucleotide or nucleoside components, which of the following regimens has been shown to be the least effective after 2 years? And you can see the 4 regimens, or the answer being none of the above, the 5th answer. Please make your choice. Okay, and the answers are a third said none of them, and the rest of the answers are pretty evenly split. So we will address this issue during the presentations. And the next pretest question: which of the following statements is true? TDF and TAF have different drug resistance profiles. The cytidine analogs, lamivudine and emtricitabine, also known as 3TC and FTC, are equally effective at treating viruses with the M184V as they are with wild-type viruses. Dolutegravir resistance mutations occur more commonly in integrase strand transfer inhibitor naive in an RTI naive and dolutegravir naive persons receiving first-line therapy. Boosted lopinavir, atazanavir, and darunavir have similar genetic barriers to resistance, and integrase resistance mutations emerge in about 1% of persons per year on first-line dolutegravir or bictegravir regimens. Once again, things are pretty well evenly distributed. So the answers, once again, to this pretest question will be addressed during our presentations. And the final pretest question: which of the following statements is true? You get a single choice. The 263K mutation is the most important drug resistance mutation for the second generation integrase inhibitors. K103N reduces susceptibility to nevirapine and efavirenz, but not etravirine, ribavirin, or doravirine. Baseline integrase resistance testing is never indicated before starting first-line therapy. The clinical significance of different fold mutations is not known. And ribavirin and doravirine have similar drug resistance profiles. One of these is true. Okay, half of the people said the answer was B, and the remaining half are sort of evenly distributed. Once again, we will cover this in our discussions. So the learning objectives of today's webinar are to describe the basic principles of HIV drug resistance mutations, explain how to understand and interpret the information presented in the guidelines update, and identify recent updates to the IAS-USA update document and how these apply to the management of drug resistance in patients. And we will be hearing about resistance to the more newly available drugs. So I will start the presentation with a description of the principles of drug resistance. So 80 years ago, Salvador Luria and Max Delbruck published a really brilliant paper describing the frequency of resistance of bacteria to virus infections with a virus-sensitive strain of bacteria. And what they showed is that about 1 in 100 million bacteria was resistant in a population of wild-type sensitive bacteria. And by showing this, they made the important observation that resistance in a microbial population is a rare event that pre-exists in that population and is not something that is induced by exposure to the population. For this work and others, the two of them got a Nobel Prize, but what they observed applies to drug resistance, antibody resistance, and so on, as we'll discuss. So virus replication results in changes, that's mutations, allowing for the virus to adapt to the presence of selective pressure. And this is true of all life forms. There is nothing perfect about replication, whether it's viruses or humans. Errors occur because we're talking about thousands to millions of nucleotides that have to be replicated. RNA viruses have a very high mutation rate, about 1 mutation per genome in a small genome, while in higher eukaryotes like us humans and our other vertebrate animal friends, that rate has to be very low because we have very big genomes, and we don't want to have to generate hundreds or thousands of offspring to get a good one. And so there are differences in the fidelity of our replication processes. And I should point out that the rates of mutation are a function of the magnitude of replication and the fidelity of the replication. And higher eukaryotes, double-stranded DNA genomes, whether bacteria or higher animals, have a proofreading mechanism that corrects some errors and reduces the mutation rate. So this is a figure that I generated for hepatitis B virologists, but it applies to all microbes. And that is the Darwinian principles of virus evolution and drug resistance, that mutagenesis is replication dependent. So you need a reproduction of the microbe The reproduction is associated with errors in the genome sequence, and that results in genetic diversity. With viruses, when somebody is infected by a single virus of, let's say, HIV, over time, there are mutations that accumulate in those viruses that are replicating, and the total population of genetic variants in an individual is called the quasi-species. The quasi-species is not a subset, it's the total genetic variance in that individual. With those genetic variants, in the presence of a selective pressure, like an antiviral drug, the more resistant viruses are more fit, Darwinian survival of the fittest, and they go on to reproduce in the presence of selective pressure. And this principle applies to monoclonal antibodies or immune responses, as we've seen with COVID over and over again. And as a result, HIV evolution in 1 year in one person equals all of human evolution over 1 million years because of the higher magnitudes of reproduction and the higher error rate. So let's get to antiviral drugs. The first one was AZT, and all we had was one drug. So we conducted a study to see whether that would benefit patients, because the first drugs tried were they all had no efficacy and some had toxicity. So mortality at the end of the study, 24 weeks, one of 145 of the AZT recipients had died, and 19 of the placebo recipients, and the study was halted. And as you can see, the various subsets in the right here of subjects all showed a statistically significant benefit in terms of survival. Because my colleagues and I were interested in drug resistance based on experience with herpes drugs, we took sequential samples from patients here in San Diego and showed that over time, the virus you isolated from patients became progressively more resistant to AZT, and that was associated with the progressive accumulation of mutations in the resistant virus. And what was observed is that over time, the patients who were on AZT started to fail as well, because the drug no longer had any substantial activity against HIV. The next class of drugs that came along were the non-nucleoside RT inhibitors, and nevirapine was the first to be approved. And what we did in our lab was look at, once we knew that drug resistance readily emerged with AZT, the nucleosides, we looked at the non-nucleosides in the cell culture and showed that the potency of the drug with multiple passages just in a petri dish, the virus became progressively more and more resistant to nevirapine. And we showed that that even after one passage in the presence of drug, you could find virus with resistance. And to prove that the mutations that we saw was the mutation, it wasn't just random evolution and mutations. There was at least initially the 181 mutation in reverse transcriptase. When you put it into a wild type virus, by molecular means conferred the same resistance. So what that does is prove two things. One is that that mutation accounts for the resistance, and secondly, that the target of the drug is in the reverse transcriptase. So in the phase 1 trials of nevirapine, what we observed was that the viral RNA dropped dramatically. This was actually the first demonstration of the dynamics of reduction of RNA, and within a week, one had a dramatic reduction of virus, and the CD4 went up very dramatically. But within 2 weeks, we could see the mutation emerge that we had observed in cell culture, and associated with the emergence of the drug resistance mutation, the RNA went back up to baseline and the CD4 came back down to baseline. So what we have learned is that single mutants are produced daily. They pre-exist, and one can find them in the wild type population even before the virus from an individual has seen drug. With great effort, one could actually find them as rare isolates, and then later, as deep sequencing became available and one could screen large numbers of viruses, you could find drug resistance mutations in the original wild type population. And these are from treatment naive patients who were infected before antiviral drug was available, so we knew that it hadn't been selected for. Double mutants are far less common. The probability of 2 mutants is the product of the 2 probabilities. And then 3 or more specific mutations in the same HIV genome, often spread in different target for antivirals like the protease and the reverse transcriptase, are extremely rare. It's hard for that many mutations to appear. And this is the principle of combination therapy. This principle was first shown for tuberculosis, where any single drug selects for resistance and multiple drugs work because of the rare pre-existence of drug-resistant mutants. So knowing that drug resistance occurs, we have learned that it is the result of drug failure. The emergence of drug-resistant virus is an inevitable consequence of the failure to fully suppress HIV or multiple other microbes with antimicrobial therapy. And it's the cause of failure because once you have it, it's a major factor contributing to treatment failure. And in the mid-90s, we showed that drugs were less effective. And some of this is because people added a single drug to a failing regimen. And then Susan Little here in San Diego showed that in patients who presented within the first month or 2 of their infection, primary infection, who had not yet seen treatment, were progressively showing more and more transmitted drug resistance. And these were data accumulated in 10 North American cities. So you will hear more about how the selection for drug resistance and the transmission of drug resistance has dramatically changed over time from Dr. Toriani. From Dr. Ceccarini Silverstein. And the Merck 035 study started in the early 90s when Indinavir came along, was the first to show what you could do with combination therapy. So the standard of therapy at that time was a combination of AZT and 3TC. And in green, you can see the dramatic impact of over a log reduction within a week, but then the gradual loss of activity as the HIV RNA went back up towards baseline. Now, with AZT3TC, you can see with continued AZT3TC, one still did not get back to baseline. And this has been shown to be attributable to the impact on replication fitness of the M184V mutation of 3TC. There is the benefit to the virus to have this mutation is that it can replicate in the presence of AZT3TC, but it's not as replication fit, as healthy as a wild type virus in the absence of drug. Indinavir had a dramatic impact as single monotherapy, but over time, but a much longer time, it went back towards baseline, and that was attributable to the higher genetic barrier and the requirement for multiple mutations to develop resistance to the protease inhibitors. And then the combination of all three resulted in a reduction to below the limit of detection, which at that time was 500 copies, and that was sustained for 2 years. So if you look at the proportion of people who are undetectable, you could see that AZT3TC didn't succeed in the long run. Indinavir got about 20-25% of people. Those were people who were 100% adherent and had very low baseline RNA levels at the initiation of treatment. But you can see that with the triple combination, sustained suppression was effective. There were some failures, and people lost to follow-up are also not counted. So what we know is that as you have increasing antiviral drug activity and the selective pressure increases, that increases the probability of selecting for resistance. But at a certain point, as you get more and more antiviral drug activity, the virus replication diminishes, and with less replication, you start reducing the probability of selecting for resistance. And in fact, if you completely suppress replication, the probability of resistance is 0. It's like putting the virus in a freezer. So with the new highly recommended regimens, when 2 second-generation integrase inhibitors were compared, you can see we're getting highly successful, this is as treated, efficacy. The people who drop out of a study and so on exceed the number of people who fail. This is attributable to more potent drugs, better tolerability, and 1 pill once a day. So one other point I'd like to make is based on the Nadia trial, and Dr. Schaefer will be telling you more about this trial, which I think is a very important trial. But the only point I want to make is that people who fail AZT, 3TC, and nevirapine, who then went on to treatment, who developed a resistance to 3TC, the question has been, what do you do with patients who are failing a regimen with these nucleosides. And the point is, if you fail with tenofovir and 3TC, going to AZT, which is not cross-resistance with the 65 mutation, really didn't make a difference. You can, the bottom line is a tenofovir 3TC or FTC regimen can be reused with a better accompanying drug, whether it's boosted darunavir or dolutegravir. And there's a reason why that is. Dr. Schaefer will discuss this study more, but the only point I wanted to make is that the 3TC mutation that results from the M184 mutation that results from 3TC or FTC hypersensitizes the virus to the activity of both AZT and tenofovir. And so you can see that there's a remarkable, more greater sensitivity of virus to, let's say, 3TC alone with no tenofovir mutations. But you even have activity with 3 or 4 mutations that are AZT associated, is close to a wild type tenofovir alone. And with 3TC 0 or 1 mutations, the 3TC resistance, zidovudine still has activity. So the point I want to make here is continuing tenofovir with a 3TC resistance mutation is a good idea. And secondly, even with drug resistance, there is residual partial activity of these drugs, which is important when one is trying to compose an optimal regimen for people who have multiple drug resistance. You may not have completely new drugs, but you have to combine even partial activities to get an optimal regimen. So to conclude, mutations resulting in resistance occur as part of a viral replication and reproduction of all life forms. Selective pressures like drug treatment or immune responses select for the most fit genetic variants. Darwin was right. Viruses generate very large populations so that all variants emerge during replication. To prevent the emergence of drug resistance, potent drug combinations are needed to treat multiple resistant variants in a large population. Drug resistance is thus both a result and a cause of treatment failure. And resistance mutations confer a spectrum of diminished susceptibility. Optimal regimens for treatment failure may often need to include drugs with diminished activity. So I will turn the microphone over to my colleague, Bob Schaefer.
Thank you, Dr. Richmond, and thank you for laying the foundation for the talks that will follow. And hello, everybody. Welcome to this webinar. I'm going to speak about the mutations associated with reduced susceptibility to the most commonly used nucleoside RT inhibitors, non-nucleoside RT inhibitors, PIs, and integrase strand transfer inhibitors, which I'll also be referring to as INSTIs. And in this presentation, I will focus primarily on NRTI and INSTI resistance and briefly discuss NNRTI and PI resistance. Dr. Cetrimini Silverstein will be speaking about the other classes of drugs, the capsid inhibitor lenacapavir, and about entry inhibitors. And I will also be referring to the nucleoside and nucleotide RT inhibitors as NRTIs. So the IAS-USA chart shown here divides the NRTI-associated mutations into those associated with reduced susceptibility to all NRTIs, shown here, and referred to as multi-NRTI resistance mutations, and those associated with reduced susceptibility to one or more of the commonly used individual NRTIs shown here. The first two types of multi-NRTI resistance mutations, the insertion of position 69 and the Q151M mutation, are rarely, if ever, seen anymore, so I won't be discussing them. The third class of multi-NRTI resistance mutations are the TAMs, also referred to as the thymidine analog mutations. The TAMs mainly affect AZT, but when they occur in certain combinations, they also reduce susceptibility to each of the other NRTIs. As shown here, 3TC and FTC have identical resistance profiles, and the two tenofovir prodrugs, TAF and TDF, also have identical resistance profiles. The NRTI resistance mutations act by two mechanisms. The TAMs shown on the left make it more likely that the chain terminating NRTI will be removed from the growing viral nucleic acid primer. As a result, they are also called primer unblocking mutations. The discriminatory mutations, several of which are shown on the right, reduce the rate at which NRTIs are added to the growing viral nucleic acid primer. The TAMs are rarely, if ever, selected by current NRTI combinations. They are primarily observed in patients with histories of ART dating back to the years in which AZT and D4T were used. However, the TAMs, because most of the TAMs are rather fit, they're also observed among transmitted drug resistance mutations, and these have been presumably transmitted from one ART-naive person to another ART-naive person over the past 1 to 2 decades. So they remain in circulation. M184V and I are the most common NRTI resistance mutations. Although they have similar effects on NRTI susceptibility, M184V is more fit and occurs more commonly. These mutations cause very high-level reductions in 3TC and FTC susceptibility. They increase AZT and tenofovir susceptibility about 2-fold, which may be why combinations of 3TC FTC with these NRTIs have been so effective. They cause low-level reductions in abacavir susceptibility. M184V and I reduce HIV-1 replication capacity, as discussed by Dr. Richmond, which may be why patients receiving 3TC FTC maintain an approximately 1/2 log reduction in virus levels despite the presence of M184V and I. And this has been observed in multiple studies, which will reference at the bottom here. But it's important to recognize that 3TC FTC is actually, these drugs are much more potent when a patient has wild-type virus, because under that circumstance, they will suppress virus levels by nearly 2 logs, or about 100-fold. Now, often virologists will speak about the fold reduction in susceptibility, so it's important for clinicians to have some idea about what the different fold reductions mean. And the range in possible reductions in susceptibility and their clinical significance differs between drugs. As shown on the previous slide, greater than 200-fold reductions in 3TC FTC susceptibility occur with M184V and I. Much lower we don't know the clinical significance of these lower levels of reduction in susceptibility for 3TC and FTC. For tenofovir and abacavir, the reductions in susceptibility rarely exceed 10-fold, but low-level reductions in susceptibility have been shown to be clinically significant, and I show that for tenofovir, clinically significant reductions in susceptibility begin at about 1.5-fold, and for abacavir at a somewhat higher level, and that's data from clinical trials that were analyzed by Monogram Biosciences. Like 3TC and FTC, AZT also has a very high dynamic range, and some viruses can have more than 100-fold reduced AZT susceptibility. But because AZT is weaker than other NRTIs, low levels of reduced susceptibility as observed with certain individual TAMs are likely to be clinically significant. This slide will summarize the phenotypic effects of the most common NRTI resistance mutations. M184VI cause high-level reductions in susceptibility to 3TC, FTC, that's shown by the thick red arrow, and low-level reduced susceptibility to abacavir while increasing susceptibility to tenofovir and AZT. K65R and N and K70E reduce susceptibility to tenofovir and abacavir, but increase susceptibility to AZT. L74V and Y115F reduce susceptibility to abacavir, and as noted earlier, the TAMs primarily affect AZT, but certain combinations of TAMs, such as the type 1 TAMs, those are the mutations of positions 41, 210, and 215, can significantly reduce susceptibility and the clinical effectiveness of tenofovir and abacavir. Much of what we know about the clinical significance of HIV drug resistance comes from clinical trials in low and middle income countries where patients with similar past treatment histories have been randomized to a new ART regimen. The EARNEST first line and more recently the NADIA trial have shown that in patients with extensive NRTI resistance, treatment with 2 NRTIs and a potent third drug, either boosted lopinavir, boosted darunavir, or dolutegravir, generally will attain high levels of virologic suppression. Paradoxically, the EARNEST and first line trials reported that patients with fewer NRTI resistance mutations actually had worse outcomes than those with more NRTI resistance mutations when they received second-line therapy. Of multiple NRTI-associated mutations. Now, as mentioned, the NADIA trial is one of the most influential trials performed in the past few years. It had a factorial design that allowed it to compare tenofovir plus 3TC versus AZT plus 3TC, and at the same time to compare dolutegravir versus boosted darunavir in persons who had experienced virological failure on a first-line regimen comprising tenofovir, 3TC, and efavirenz. And it had 464 participants. At baseline, 87% of patients had M184V or I, and 50% had K65R. The first main finding was that dolutegravir was non-inferior to darunavir at attaining virologic suppression after 1 year with virus loads less than 400 copies in 90% of patients and less than 50 in 80%. The second main finding was that at 1 year, tenofovir and AZT were equally effective, despite the fact that K65R, which reduces tenofovir susceptibility but increases AZT susceptibility, was present in 50% of patients. And at 1 year, 4 patients on the dolutegravir arm developed INSTI drug resistance mutations, but no patients on the darunavir arm developed PI resistance mutations. Now, the week 96 data added some new information, and at week 96, it was shown, first of all, that tenofovir became superior to AZT, as it was associated with a virus loads less than 400 copies in 92% versus 85% with AZT. And it also at 2 years, having received tenofovir was associated with reduced emergence of INSTI resistance. So by week 96, 9 subjects who received dolutegravir developed INSTI mutations, but 6 of those patients were in the AZT arm, and they developed much higher levels of cross-resistance to the other INSTIs, while only 3 of those patients were on the tenofovir arm. So paradoxically, tenofovir did better than AZT, but that's likely because tenofovir is a more potent drug than AZT, and AZT also requires twice daily administration. The INSTIs can be divided into the first generation inhibitors, raltegravir and elvitegravir, and the second generation inhibitors, dolutegravir, bictegravir, and cabotegravir. Dolutegravir and bictegravir have much higher genetic barriers to resistance than the first generation inhibitors based on data from in vitro passage experiments, clinical trials, and in vitro susceptibility testing. Dolutegravir and bictegravir also have higher genetic barriers to resistance compared with the long-acting INSTI, cabotegravir. Mutations at position 148 lead to the highest levels of dolutegravir and bictegravir resistance, but only when they occur with additional accessory mutations. Usually 2 mutations will lead to very high levels of resistance to these drugs, and of course, cross-resistance to the whole class. R263K was initially the most commonly reported mutation in previously INSTI-naive persons receiving dolutegravir. Alone, it reduces dolutegravir susceptibility about 2-fold. However, G118R is increasingly being reported in INSTI-naive persons receiving dolutegravir. Alone, it reduces dolutegravir susceptibility by more than 5-fold. However, it is associated with reduced replication capacity. N155H has also been reported in a small proportion of INSTI-naive patients receiving dolutegravir, but alone, this mutation does not have much of an effect on susceptibility to the second generation INSTIs. As shown in the figure here, many of the integrase mutations that cause high levels of resistance to the first generation INSTIs, raltegravir and elvitegravir, cause very low levels of cross-resistance to dolutegravir and bictegravir. In addition, INSTI DRMs have almost never been reported during the several thousand patients who've received first-line dolutegravir or bictegravir in clinical trials. Based on data from the Viking clinical trials, however, we do have some information on the clinical significance of fold reductions in susceptibility, at least to dolutegravir, because in that clinical trial, there were patients with drug resistance from previous raltegravir or elvitegravir use that went on to receive dolutegravir. And a 4-fold reduction in dolutegravir susceptibility was associated with the beginnings of a reduced virological response, while a 13-fold reduction was associated with an absence of virological response. It's important to recognize, though, that the clinical cutoffs that I showed earlier for tenofovir and abacavir, and these cutoffs, and ones I'll show for protease inhibitors, they're very approximate. I think they give some context about what different fold reductions in susceptibility mean, but they're generally derived from a single clinical trial, so they're definitely not absolute cutoffs. This slide shows data from 5 randomized clinical trials in which NRTI experienced but INSTI-naive subjects were treated with a dolutegravir-containing regimen. INSTI resistance mutations emerge, as shown here, in about 1% to 4% of subjects by week 48 or week 96 in these trials. The proportions with emergent INSTI resistance mutations were higher in the trials from low and middle income countries in which genotypic resistance testing was not used to choose the drugs administered with dolutegravir. So in the Sailing study and Dawning study, somewhat lower rates were observed because in those studies, resistance testing was used to help guide therapy. Each of the 29 subjects with emergent mutations had one or more mutation at positions 118, 263, 148, or 155, the mutations we mentioned earlier when reviewing the mutation chart. The figure on the left here shows that in the US, transmitted INSTI resistance mutations occurred at a prevalence of about 1% in 2018. And more recent data that Dr. Setcherini-Silberstein shared with me that is about to be published from Europe showed that these very low rates of transmitted INSTI resistance in Europe have persisted even through years 2019 through 2021. As a result, routine integrase sequencing is not recommended for ART-naive persons. However, it is recommended for persons who previously received PrEP with cabotegravir, and I believe that it's also recommended for people considered to be at high risk of transmitted resistance, and I believe that that category should include people who have mutation who have demonstrated transmitted resistance with the M184V, K65R, because they're pivotal to most first-line regimens, as well as those with multi-class resistance. Moving on to the NNRTIs, this slide shows that the NNRTIs, nevirapine and efavirenz, have similar resistance profiles. The most common drug resistance mutations associated with these NNRTIs are K103N, Y181C, Y188L, G190A&S, and in subtype C viruses, V106M. Etravirine and rilpivirine have similar, have resistance profiles that are similar to each other. Etravirine, however, has a higher genetic barrier to resistance and often requires 2 or more mutations before complete activity is lost. Y181 mutations are important for both etravirine and rilpivirine, while Y188L is important for rilpivirine. E138 mutations are particularly and probably uniquely relevant for rilpivirine. Doravirine has a unique resistance profile. Alone, Y188L is associated with the highest levels of reduced susceptibility. Several pairs of mutations that include either a mutation at position 106 or a mutation at 227 with one other drug resistance mutation are associated with very high levels of reduced doravirine susceptibility. However, despite the fact that NNRTIs may have different resistance profiles, there have been few examples in which NNRTIs have been used to salvage therapy in patients who just failed a previous NNRTI-containing regimen. And that's because in patients receiving NNRTIs, multiple NNRTI resistance mutations often develop, and because combinations of 2 to 3 or more mutations that are seen during acute failure with an NNRTI, there tend to be higher levels of cross-resistance from what we see when a patient just has a single NNRTI resistance mutation. The 3 commonly used boosted protease inhibitors, lopinavir, atazanavir, and darunavir, have high genetic barriers to resistance. In fact, most patients with virological failure on an initial PI-containing regimen do not develop PI-associated drug resistance mutations. because the three most commonly used boosted PIs have been used to treat patients who failed other PIs in the past and have PI resistance mutations, there are some data on the clinical significance of different fold reductions in susceptibility. Based on the POWER trials, it has been estimated that a 10-fold reduction in darunavir susceptibility is required before there's a diminished virological response compared with wild-type virus, while a 90-fold reduction is required before there is a complete loss of virologic suppression. And a 90-fold reduction usually occurs from the development of 5 mutations among those shown in the chart here. Usually it's 3 of the major mutations shown in bold and 2 accessory mutations. Boosted lopinavir and boosted atazanavir have lower clinical cutoffs. For atazanavir, there has only been enough data to calculate a single clinical cutoff, and so reduction in susceptibility begins when reduction in clinical activity begins when the susceptibility is reduced more than 5-fold. The most recent experience with PI resistance comes from second-line therapy in low and middle-income countries where patients receive boosted lopinavir in combination with NRTIs that are often compromised as a result of resistance that developed during initial therapy. This figure from a systematic review shows that PI resistance mutations are uncommon in studies that looked at patients after 1 year of therapy. But in studies that looked at patients after 2 years or longer of therapy, emergent PI resistance reaches about 20% of patients. The risk of emergent PI resistance in PI-naive persons receiving boosted darunavir is much lower than for lopinavir. It occurs at an extraordinarily low rate and to some extent is almost reportable. Fewer data are available for boosted atazanavir, but it's likely to be similar in the rates of resistance that we see with boosted lopinavir. So I will stop sharing now, and because this concludes my talk, and hand it over to Dr. Setruti-Silverstein.
So thank you for the invitation, and I will focus, as Dr. Richman and Schaefer said, on the resistance, particularly to the new HIV drugs. So just I would like to make a summary of what we are today. We experience an evolution of antiretroviral therapy. So in the beginning of the early 80s, we didn't have a treatment, but as Professor Richman said, after the late 80s, we were able to treat the patient with zidovudine monotherapy, and after with sequential NRTI monotherapy and the dual NRTI therapy. And at that time, the drugs were with low tolerability, and the clinical aspect that the clinicians were really addressing is to avoid the death and the opportunistic infection. And the target of the antiviral treatment was efficacy and effectiveness. After, with the advent of the discovery of the first protease inhibitors, we could start with the heart, and but still we were having a high number of pills, and we start to have as a clinical complication lipodystrophy and not so good quality of life yet. But the goal, the target of the treatment was now the durability. And in the early of the 2000, we were starting also to work on deferral of therapy. But on the late of the 2000, with the start trial, we saw how it's important to treat the patients as early as possible. So we started the early initiation of therapy, and we started to have a few pills, particularly the single tablet regimens. And now the clinical target issue, particularly, is the aging and comorbidity. And the target, of course, is sustainability. And finally, where we are now. Now we are with the rapid heart, test on treat. We try to treat the patients as soon as possible when everyone an individual discovered to be HIV infected. We start to have treatment that we can use as a long-acting. And so which are the clinical aspects now? Today is the frailty population, and we are as a target a lot of studies on eradication. So in this period, we have some new drugs, particularly why we need new drugs, particularly for patients that have multi-drug resistance that previously failed the treatment. But not only, of course. So I will focus particularly to some of the drugs that have been recently approved. And why first to say we have multi-drug resistance people that are frequently highly treatment experienced, but not all the highly treatment experienced patients are multi-drug resistant, we should remember. But normally the people that have multi-drug resistance are heavily treatment experienced, people living with HIV, because they have the previous story of zidovudine monotherapy, the sequential monotherapy. So, and on that, all of this reason, how Professor Richman elegantly showed you before, and particularly if the patients, even if it was highly adherent, but after they were not managed to fail, they managed unlikely to fail because the treatment was not so potent, we have a lot of development of resistance. Now, of course, we have early initiation of therapy with better treatment, with higher genetic barrier, and more potent. So today we have less selection of resistance virus. So this is very important. But the multi-drug resistance, so are normally older people that have been treated in early days of heart. And also we should not forget about younger people that acquired the infection with a congenital by vertical transmission, and particularly also individuals that the transmitted drug resistance has been occurred. Why is important the multi-drug resistance? Because we know, and for the sake of time, I cannot show, but still in our study that we currently did in Italy, still the patients that have multi-drug resistance are at high risk of death. At that time, of course, we didn't have yet the new drugs like darunavir, maraviroc, and particularly altegravir, that was the first class that saved these patients. So the multi-drug resistance is an issue when we have the patients. And here we can see how this is in line what I said to you before. So this is, we are doing resistance tests for many clinical centers, and you see here more than 30,000 isolates, how we see the changing scenario of the resistance over time. So from the beginning to 2010, there is clearly a decrease of resistance. You can see the white bar that is failing without resistance, that is increasing from 15 to 40%. But after this is stable now around 60%. And so we have around 40% still from 2010 that have persistence of resistance, and some patients, you see, around 5% with at least triple class resistance. And of course, we see an increase of the integrase resistance, and the patients that fail with integrase resistance are particular, you see, 34% when have 3 classes resistance. Why this is important? And you know that Bobo Schaefer is driving and is analyzing and supporting all of us, providing the Stanford database that we allow with this system to calculate the genotypic susceptibility score. So each of us, if we have a virus that is resistant, we can see if the drugs that we provide together with the treatment that we want to provide is they could be active. And so even if this slide would show us that even if a patient have a triple class resistance and was given after the resistance test a full regimen, the patients did not achieve less, achieve a probability of virological success 100% after the previous failure, while patients that have triple resistance and a poor regimen, so not all the regimens were really fully active, you see that the probability to reach the virological success was 62%. Overall, there was 87% of the patients after 1 year, they achieve virological success after a previous failing. So I will start to speak a little bit about these new drugs, and very important, of course, is as a new mechanism of action is lenacapavir, that in this moment is granted to use for European Commission in UK, and that is submitted by FDA to FDA for approval. And now we can use particularly for treatment of adults with multi-drug resistance infection, where there is no possibility to use to have too many other active drugs. But of course, this is a very interesting, very potent in vitro with picomolar potency, and is the first-in-class capsid inhibitor. And of course, since it has a different mechanism, is sensitive to all HIV drug resistance virus with all the classes, and is active against CCR5 and CCR4 tropic virus. And what is the peculiarity? That this drug bind the capsid and allow with the 3 different mechanisms to inhibit the viral infection. First, inhibit the nuclear entry of HIV DNA that is mediated by the capsid, and after, of course, the viral assembly of the capsid, and finally the assembly and the virus assembly and the release. And this drug we can use as a oral administration, but also subcutaneous every 6 months. And so it's been used now so far for treatment, and they are starting also to think to do also as a prevention. Here, what we see, how this drug was able, in the first graph, you see in the randomized group, so there was people that were for 14 days with a placebo, so containing the failing regimen, and the people that receive lenacapavir. And you see that there was a very big decrease of from baseline viral load on these individuals. And the efficacy was maintained in both cohort, non-randomized and randomized cohort. So overall are 72 patients, and you see how there was a good efficacy here after 26 weeks, also with the recovery of CD4 cells. And here are the new data that have been presented with just about the randomized cohort. And you see that at week 52, excuse me, there was still a very good maintenance of viral suppression, even if it's minus 50 or minus 200 copy. And the efficacy was changing, of course, to the active agents on optimized background regimen. And you see that, of course, if there was no full active agents, 0, you have 67%. So still 4 out of 6 responded, while if you have more than 2, of course, 94% of activity. So very important. But like all the drugs, if if a drug is active against the virus, we know, and Professor Richman showed us, there is a development of resistance. And so also in vitro, they were testing different assay, you can see here with the passages, and we saw the development of 7 mutations at 6 particular amino acids. And so, of course, the new of our list of the mutation, we describe for the first time the mutation that are associated with lenacapavir. And the first have been identified in vitro, and particularly you can see that 2 of them, M666I in combination with the 67H, can have a very strong fold change of resistance. And also, interestingly, that normally, if there was this resistance anyway, you see that the virus did not replicate very well. So you have a very low replication capacity. An important message is that we don't need so far to do the baseline test resistance for lenacapavir, because there is, even if the capsid is quite polymorphic, but there is no natural resistance. These are two studies published with 1,500 individuals that they did in France, and in a recent paper in Cameroon, in collaborating with us, they retrieved more than 2,000 sequences of drug-naive individuals. And you see that according to the different subtypes, there are some polymorphism, but indeed there is no, very rare, less than 1%, 0.1% about resistance in naive patients, so that have not been treated. Of course, when in the phase 1b study, the post-monotherapy analysis reveal the emergence of low-level resistance Q67H in 2 participants. Particularly, you can see with the low doses of lenacapavir. And this is interesting because this mutation, as you can see here, has the better replication capacity and also low resistance. So in some way, was this the reason that probably was selected? And in the patient that fail the Capella trial, so the patients that have been treated with the multi-drug resistance virus, you see that there are some patients that fail. The majority are failing with the M66I, that is the mutation that is very important also in vitro. And the majority of the mutation that you can see have been previously identified during the selection for in vitro resistance. What is important that so far the patients did not emerge new resistance to the other component of the optimized background therapy. So about the genetic barrier of these drugs, particularly when is functional monotherapy, you can have the risk to develop the resistance, but not all the patients fail with the resistance. This we should say. And another important possibility to use lenacapavir, as I told you, is also in the drug-naive patients. So the long-acting is a very interesting approach. And here you can see the long-acting with lenacapavir that has been used with FTC TAF, that after have to switch to TAF or bictegravir, or lenacapavir oral with the oral administration, or with the comparison bictegravir FTC TAF. So you see that there is a very good virological suppression maintenance at 28 weeks. There were 2 failing patients with the resistance, and again is the same mutation 67 and together with the 70R. So we start to see the few patients that fail, sometimes of course they fail with resistance and with the mutation that we know. So again, speaking about multi-drug resistance, why we need these new drugs? Because they could, you see here, have an efficacy, and you see how here there is the first endpoint of all of these new compounds that have been approved for treating patients with multi-drug resistant virus is normally at 1 week or 2 weeks to see a decay of HIV RNA. And you see how ibalizumab, stemsavir, and lenacapavir have a good efficacy. So now, speaking about another drug that has been already approved by FDA in 2020 as a first-in-class oral attachment inhibitor that binds to the GP120, here we can know that there is no consensus yet about the resistance mutation and pattern. So for us, we didn't put a signature on the bars on our list, but we know that particularly some subtypes, there is some heterogeneity of activity, and with subtype AE, you see that there is no activity. So we in some way recommend for the country where there is a more prevalence of this recombinant AE to do the subtyping, could be important for usage. And so far, there is, in the NRS, there is a list of the 4 positions that have been associated with the resistance in vitro. and indeed, two of them, the 375 and the 475, are very frequently in the recombinant AE. Some of them are little present also in subtype B. So we need to know, and but today we don't have yet a correlation of baseline resistance and activity of the treatment. But what I wanted to show you here, that seems that the best way to find predictors of efficacy of this with fostemsavir with the high treatment individuals is when you combine a susceptibility combined by genotypic susceptibility score and the phenotypic susceptibility score, but not for all the drugs, but specifically when you are using new usually drugs. And so here you see indeed with the red dash that of course are less people, 70 patients, but you can see clearly a better efficacy respect to individuals that have no active drug at all. The other drug that I want to mention is ibalizumab. That is first human monoclonal, humanized monoclonal antibody that can bind to CD4 when CD4 is already bounded to GP120. And the GP120, particularly, we know that there is a specific region in the V5, that is the variable region 5 of the GP120 that is rich of glycans, are essential for the binding and the blocking. And so as a resistance, so just to say that the glycosylation is a very important process for which the virus try to evade the immune system, because in this way there is less neutralization and so prevention of immune recognition. But the glycans can play also a role in virus cell attachment. And we know that losing some asparagine-linked glycosylation because there is less functionality activity of ibalizumab. And so here is just an example that indeed the patients that failed fostemsavir develop resistance, but also you can see here he lost one glycosylation site, and particularly more close to the N-terminal part of the GP120 that seems to play a major role of resistance. This is very complicated algorithm, but just to say to you that they are working. This is made by the Max Planck people, bioinformatician, to see that indeed is a very complex if there is more glycosylation, and particularly all the two in the V5 loop, there is very highly sensitive to ibalizumab. If there is just one, still, if it's maintained in N-terminal, is more active, is in another position, if it's depending how is the size of the view2. So just to say to you how it's complicated to do so far about resistance for ibalizumab, and indeed we need to do the sequencing of the GP120. Finally, I want to speak one moment about another drug combination that is very useful for as a long-acting, that is rilpivirine with cabotegravir. And probably you heard that there was some, so this combination is very active. There have been 3 phase 3 trials that allow the using of this combination in individuals that are suppressed therapy, virological suppressant, so they can be switched to this combination treatment. And we know that there are several factors that can be investigated. In the beginning, the first failure were individuals who had have tri-ration in a specific flare study that have the A subtype and particularly L74I mutation. So there was a lot of discussion and about this. So in this analysis, they analyze all 1,039 participants that were really naive to cabotegravir and rilpivirine, and they analyzed the only 13 virological failures that have been presented here, and they try to understand which were the baseline factors that have been associated. So the most important was the rilpivirine resistance at baseline, but also after the baseline HIV-1 subtype A6 or A1, that now we know that there was probably more A6, but still, because in Russia there is a lot of prevalence of A, and before was thought that it was A1, but now we know that it's A6. And so there is, we know that at least 2 factors between pre-existing rilpivirine resistant mutation, the subtyping, or also very important, because these are injectable antiviral regimen, is the body mass index. So if you have at least 2 of these factors, you have higher risk to fail the treatment, even if it was very successful. You see just 1% of participants have a virological failure. So there was big discussion about this subtype, and A1 and A6. So very elegantly, the group of Daniel Kuritzkes show at CROI that indeed in the A6 virus, when there was the mutation 74L, there was, the L74I polymorphism conferred a great replication to this virus, and particularly when there was also the mutation that are important for resistance, particularly for also for cabotegravir, like 118, 140, 148, and 263. So there was, you see, an advantage in absence of drug, but also in the presence of cabotegravir. So in some way, these findings show that this polymorphism can provide indeed a better replication that can be associated with the virological failure to this regimen long-acting. So here it has been presented also at CROI the few resistance cases of the participants that have confirmed virological failure in the ATLAS studies. And you see that in some cases in the red, you have the emergence of resistance that have not been previously. And you see that sometimes there is also the N155H. And this is the reason why also there is this mutation also for cabotegravir. So there are the classical integrase resistance mutation and the classical that we know rilpivirine mutation. Finally, just to say that it has been presented now at Glasgow. Again, they try to understand which were the factors associated to the failure of this long-acting. And in this moment, of course, is very advantageous. I don't know how is the use you are practicing this usage, but in Italy we are approaching now. So it's a very important possibility to treat with this long-acting event also in the future. And here what they saw at the end, finally, that indeed the best model was again that if you have no one of these factors, rilpivirine resistance baseline or HIV subtype A6A1 or BMI more than 30, so if you have no baseline factors, you have the very low risk of failure. If you have at least 1, you have 2%, but if you have at least 2 or more, you have 19% of failure, you see. And you see that there is a very quite good sensitivity of 48% and specificity 96.7%. So if we want to summarize, we know that HIV resistance is always evolving. Also, when we use more the compounds and when we have new drugs, of course, we need to learn the new sequence, the new resistance scenario. And so if we are speaking about these 3 new drugs, ibalizumab, fostemsavir, and lenacapavir, we need to think that we need to sequence probably all the GP120 if we want to understand the resistance for ibalizumab and fostemsavir, while for lenacapavir, we need to sequence the capsid that before we didn't do. And about the genetic barrier, we know that ibalizumab for stemsavir is very low. For lenacapavir is low medium, we still need to identify. And we don't have yet a real genotype interpretation for ibalizumab for stemsavir, and we surely need more learning from phenotype. And also we start to have some data about lenacapavir, but still we need to learn more from phenotype. So in conclusion, we can say that the resistance testing remains surely a cornerstone of the treatment, and the resistance is crucial for the prevention and management of failure of the treatment. And overall, we can see a decrease of HIV drug resistance at failure, but also as a transmitted resistance. This is very important. We still have a few patients that harbor multi-drug resistance that are at high risk of death. And so this is the reason why we need the surely novel mechanism of antiviral drugs that can play a critical role for such a proportion of these individuals. But after, new mechanism of action can be very good for all the patients, and particularly some new drugs with a long-acting strategy can be really very useful and can change the scenario for treatment and thinking about prevention. If you can provide one injection every 3 or 6 months, of course, make a big difference. And so the evolution of HIV resistant profile follow, of course, the improvement in the regimen setting. So we need always to be updated. So I conclude acknowledging this is a proverb, African proverb that I like always to finish my presentation, that if you want to go fast, go alone. If you want to go far, you go together. So now.
So thank you very much, Francesca. That was very informative. I think I want to apologize for the group. The three of us had more we wanted to say than we had time for. And why don't we go through the post-test questions, and then we'll figure out how to deal with the Q&As. We've tried to answer some of them by text. So the first post-test question: after failing a regimen of tenofovir, lamivudine, efavirenz, which of the following regimen was shown to be least effective after 2 years? Single choice. Okay, well, Bob Schaefer did show the slide that the AZT regimen was clearly inferior to the tenofovir regimen. So the answer is C. And why don't we move on to the next question? I should mention that the Nadia trial, the year 2 results showed the inferiority, and it was in a Lancet HIV paper. Which of the following statements is true? And please read them for yourself and answer. The correct answer to question 2 is C. And the last question. Which of the following statements is true regarding the drug resistance mutations? The correct answer to question 3 is B. We have gone over, and I apologize, but I want to thank people for attending. And the question is, Donna and Jose, is there a way for us to answer questions offline after the termination of the webinar?
Yes, we could do that, but if the panel has the availability, you're free to stay on and answer a few questions.
Okay, well, if people would like to stay on, we will make our effort to answer the unanswered questions.
Doug, I think that would be the best, because also those questions and answers will be captured in the archived webinar, whereas the written answers will disappear once everybody leaves. So as long as you, Francesca, and Bob can stay on, please do answer as many questions as you can.
Okay, why don't we try to answer them briefly in the interest of time? So Bob, the first question, why did we remove D67N as a thymidine analog mutation?
Well, we still have it for AZT, and I'm not sure of the discussions that went on. It's definitely not as important as the type 1 TAMs of 41, 210, 215, especially 215Y and F are definitely the most important. So I think we have it for the D67N. That's we have it for zidovudine, and for the multi-NRTI resistance, we've emphasized the 41, 210, and 215.
Okay, I will quickly answer the next question about what percentage of adherence various classes of art result 50 or 75%. In general, once you get down into the less than 50 or less than 75%, you end up selecting much less for resistance because the selective pressure has been missing, but there are differences among each drug and each class, largely on the basis of the half-life of the drug and the genetic barrier. And so a drug with a longer half-life, like efavirenz or 3TC, tenofovir, will select for resistance more readily than something that doesn't last very long. And so it really varies by drug, and you can just answer these on a population level. The bottom line is to eliminate poor adherence as much as possible, which long-acting drugs may do. Is there any definitive evidence about when or if to get an archived genotype? Bob?
Well, this is an emerging area, and of course, in people who are virologically suppressed and you want to modify therapy to simplify it, to avoid drug toxicity, drug-drug interactions, or improve adherence, you want to change therapy, but you don't have anything to go by because you can't test the person at the time. But I think there are two ways to think about it. First, you can look at the regimens they were on in the past, and if they were followed very closely, and had plasma HIV resistance testing at the time of each virological failure, then you don't need to do any additional testing, and you probably could use those results. On the other hand, if you don't have any data on the patient, then it's believed that a PBMC genotypic test may be useful, especially if you see mutations that are present. You would avoid drugs that would be affected by those mutations. But if you don't see mutations, you would still have to look at the person's past medical history to decide whether it's likely they would be susceptible to a drug. So I think there are some circumstances where it is worth doing the test, but there are many circumstances in which you could go solely by past tests and treatment history.
Okay, I will quickly answer the next question because it was asked before Dr. Ceccarini-Silverstein's presentation. In terms of the duration to select for INSTI resistance, the integrase inhibitors, the second generation ones, look like they're selecting at about 1% per year, at least with dolutegravir. And bictegravir, it's really a case report or two over the last 5 or 6 years, if I'm not mistaken. So it really has to do with probably poor adherence. The next one, are there data regarding the magnitude of lower resistance barrier on cabotegravir compared to the other second generations? I can just make one quick remark that a number of these mutations have a bigger magnitude effect on the loss of susceptibility to cabo than they do to dolutegravir and bictegravir. But Bob, do you have anything to add to that?
Yeah, that was going to be half of my answer, and the other half was that during in vitro passage, more mutations occur to cabotegravir than dolutegravir and bictegravir. But in clinical practice, the drugs that cabotegravir is used only essentially in patients without active virus replication, because it's used for PrEP or in combination with rilpivirine for suppressed patients. So we really don't have clinical data regarding that.
Let me go to the question about K65R and M184V, the issue of the combination, and then there was a later question about whether they can actually occur in combination.
I answered that. I said that those
Okay.
two could occur in combination. K65R, even though they both reduce virus replication, when there's sufficient selection pressure from the use of tenofovir and 3TC or FTC, then we do see them in combination on the same genomes.
And
I can reply if you want. Yeah, for the cabotegravir rilpivirine treatment, how can we know for certain there are no baseline rilpivirine mutation if the patients who are eligible to switch must be suppressed and therefore difficult to genotype? So normally we do, surely in Italy, we try to do the resistance on PBMC. But again, if there was no treatment before with an NRTI, but you cannot exclude that was transmitted a resistance as a drug-naive individual with an NRTI that is particularly in the previous year was around 10%. So normally it's better to do the resistance at DNA level, yes, on PBMC.
But that's, I mean, in the US, I mean, and according to, I think, the FDA approval, I don't think it's standard. But if somebody is particularly, paranoid about that there might be transmitted resistance before starting therapy, then I guess you could do the genotyping. But I would say we don't do it routinely in the US.
right.
Particularly if you have not been treated with an NRTI before.
Right, of course, yeah. If you haven't been treated before, I'm sorry.
Okay, the subtype C resistance, that was answered.
No, I said I would answer that. So is this the one that said, is HIV-1 subtype C
More
resistant?
frequent.
Is that it?
Yeah.
Is HIV-1 subtype C resistance to tenofovir occurring more frequently? Are there other non-B subtypes which are more prone to develop drug resistance? I think one of the things that each of the subtypes has a different predilection for developing certain mutations. And subtype C viruses were more likely to develop K65 during in vitro passage experiments and to some extent in patients with virological failure. But I believe nobody has shown that K65R that patients with 8 subtype C viruses are more likely to fail on a tenofovir-containing regimen. But if they fail, they are slightly more likely to have K65R. And I think that same principle applies to all the different subtypes. We really don't see different subtypes doing worse on different regimens, but we do see that different subtypes may select preferentially for certain mutations. There are probably about 7 or 8 instances of that. And in some ways, it's sort of trivia to know those instances. I guess one of the most well-known ones is efavirenz, and V106M is much more like, V106M is much more likely to occur with efavirenz. But there's no data that subtype C viruses are more likely to fail when they receive an efavirenz-containing regimen.
Okay, let me just quickly answer the question about HXB2, which is a lab strain. The data that we are describing are really based, and the guidelines figure are based on isolates from patients and multiple different large numbers, so that the lab strain data are only used for site-directed mutagenesis to confirm the mutation. But and then moving on to the last question about hepatitis B, I'll let Francesca expand, but let me just say that one critical thing about Cabernet is it does not contain a component that has activity against hepatitis B. And so it's absolutely a bad idea to administer that drug to someone who is hepatitis B antigen positive. And it's been shown that now more recently that even very low levels of hepatitis B DNA are associated with hepatic disease progression. So it's really essential to get hepatitis B serologies before considering a regimen that does not contain tenofovir. Do you want to expand on that, Francesca?
Yeah, no, I fully agree. And also, we know that not only with the chronic really hepatitis B infection active, but also only the anticore positive, for instance, that we think that is a resolved infection. We know that if you don't treat the patient with FTC or tenofovir, we can see a little bit. So it is important that whenever there is a chronic infection of hepatitis B or previous infection in some way, so anticore positive, that probably is not better, is not good to use this regimen. I agree.
Okay, well, thank you very much. I want to thank the attendees, the hundreds of attendees who attended. We appreciate your interest, and it's been a pleasure presenting what the information that we're really interested in talking about. So thank you. And Jose or Donna, do you want to?
Yes, and again, we'd like to thank our presenters for very informative and thorough presentations and discussion. As a reminder to our audience, evaluations and how to claim continuing education credits will be emailed by 5 p.m. Pacific time tomorrow. This will enable us to view all of those that have attended today's live broadcast. Again, access the on-demand or the updated 2022 drug resistance mutation on the IAS-USA website. Tomorrow we have an upcoming course on the IAS-USA guidelines regarding the antiretroviral drugs for treatment and prevention of HIV infection in adults. And that course coincides with the release of the recent guidelines, which are now published in JAMA. And here are a list of upcoming webinars. Here are both of these webinars are scheduled for January of next year. Again, for more information, please visit our website. Dialogues will be scheduled for the coming year. So again, please stay tuned on our website. And last reminder for CROI, that registration is now open. And please visit the CROI website for additional information. All right, and again, thank you to our presenters and for the audience for your attention. This concludes today's webinar.
Thank you.
Thank you.
Bye-bye to everyone. Bye-bye. Thank
Bye.
you for the invitation.
Arrivederci. Buonasera.
Arrivederci. Ciao, Doug. Bye-bye, Bob.
Bye.
Everyone.
Bye, Francesca. Bye, Doug.
everyone.
Bye,
Thank
Jose, Donna. Thank you so much.
Yes, thank you, Donna and Jose. Thank you very much. Bye-bye.
Thank you so much, everyone. We'll let you know when the archived webinar is up. Lots of great comments and gratitude from the audience. Bravo. See you soon.
Bye-bye.
Bye.
See you soon. Bye-bye. Thank you. Bye-bye.